Active data biology has multiple views of data sets, each providing a different set of interactions for the user to visualize patterns and browse their data. The pathway view allows users to view project data within the context of biological functions, as it layers data directly onto images provided by KEGG, representing molecular pathways for metabolism, genetic information processing, environmental information processing, etc. For this tutorial, we will be showing our metatranscriptomics dataset from a simplified natural community. This experimental dataset has some unique features which make the pathway exploration especially informative. Because the dataset comes from a simplified natural community, there are many different organisms present. This particular cyanobacterial consortium has 13 heterotrophic bacteria living within the cyanobacterial phototroph. Thus, any specific gene in a pathway could have expression data for any member of the consortium. The second feature is that this is a time course experiment with 4 samples across 28 days to observe community succession and dynamics. The pathway view can be accessed from both the Heatmap and the canvas. Today we'll go straight to purine metabolism by clicking the pathway button and searching for the purine biosynthesis pathway. In the pathway map itself, there are several different graphics, each representing a biological entity. The boxes represent gene products, most commonly measured as proteins or mRNA. Circles represent metabolites or other chemical compounds. The rounded rectangles represent other pathways that tie into the one currently shown. An image legend provided by KEGG is found in the Help or Pathway menu. The color shading of the boxes conveys the quantitative information from the heat map. Reddish boxes show high abundance. Bluish boxes show low abundance. Although the underlying data is from a community of organisms, the pathway is colored from only one organism at a time. You can change which organism is shown in this drop-down box. The name which appears in this drop-down is taken from the metadata file and is customizable by the user. For starters, we'd like to know how expression in this pathway changes for one of the heterotrophs, Halomonas strain HL93. So let's select that from the drop-down box. Data are initially shown for time 0, or the beginning of the study. The blue shade of most proteins at this time point indicates that expression of this pathway is generally low. For time course datasets, the pathway view has the animation controller to display the variations of expression across the time. By pressing play, you can see the expression of each transcript for this organism change over time. We note that most genes in the pathway turn from blue to red in the second time point, showing that this pathway is significantly upregulated by day 7 of the experiment. To investigate a single gene compare expression across all members of the consortium, simply click on the shaded box. We'll choose PRPP synthase for this example, EC 2.7.6.1, near the beginning of the purine synthesis pathway. The expression of this gene is nearly constant for all organisms in the consortium, except Halomonas HL93, which has a unique pattern. Further down in the pathway, we'll choose PureH, or IMP synthase. We note that it shares the same unique pattern of expression within the consortium, with Halomonas being the only community member with a change in expression abundance. You can save the protein to the canvas by pressing the Pin button at the top of the window. Alternatively, you can save a copy of the image to your desktop. A second view of the data is shown below the pathway image, which shows gene expression with explicit phylogeny grouping, drawn from the metadata file. The pathway view puts your data into the familiar contexts of biological functions. Together with the rest of the active data biology application, it will enable you to interact with and explore your data in new ways.